prevention is better than cure, and especially when there is no cure. This particular hearsay can be very well associated with a severe viral disease. Yes, I am talking about AIDS, acquired immunodeficiency syndrome. Hello everyone, this is Dr. Madhushri Chatterjee and welcome to MC Biology classes. In today's session, I'll be talking about AIDS, its causative agent, that is HIV, human immunodeficiency virus, HIV's replication cycle, and some misconceptions or myths associated with this particular disease. So yes, without wasting time, let's get started. See, the term AIDS itself is self-explanatory. Why? Because the term itself signifies all the features associated with this particular disease. Let me start with the first letter, that is A. A stands for acquired. Acquired. That means this particular disease someone acquires during his or her lifetime. It's not congenital. Okay, that is point number one. When I stands for immunodeficiency. It stands for immunodeficiency. What does it signify? It means that this particular disease weakens the person's immune system. Okay, that's why it's called immunodeficiency. And last but not the least, S. It stands for syndrome. S stands for syndrome. And the term syndrome means a group of symptoms. Okay, so this is all about AIDS. And now the next thing that comes in mind is, what is the causative agent of this deadly disease? Yes, this disease AIDS is caused by a virus which is known as HIV. Okay, and the full form of HIV you all know is human immunodeficiency virus. Let me write it here. It's human immunodeficiency virus. Now, what is the special feature of this virus? Is this virus same as all other viruses? No, it's not. Speciality of this virus is that it is a retrovirus. This is a retrovirus. Now, why do we call it a retrovirus? See, in case of HIV, the genetic material is single-stranded RNA. Okay, it's single-stranded RNA. But it's not like that any virus possessing RNA will be referred to as retrovirus. No, it's not that. RNA is present as genetic material, fine, but along with RNA, a particular enzyme is present, and that enzyme is known as reverse transcriptase. Reverse transcriptase. So, a virus possessing RNA as the genetic material as well as reverse transcriptase enzyme. Why it is so special? Because this reverse transcriptase enzyme will carry out reverse transcription process. What is that? See, you all are well acquainted with central dogma. What happens during central dogma? DNA gets transcribed to RNA, and from RNA, protein is synthesized by the process translation. So, if an organism, say if a virus possesses RNA as the genetic material, what will happen? First of all, it has to be reverse transcribed to DNA, okay, and then from that DNA to RNA and then protein, the process will be like this. So, since this retrovirus possesses single-stranded RNA as the genetic material as well as this particular enzyme for carrying out the reverse transcription process, we call it as retrovirus, and this is special about HIV. Now, how does HIV infect a person? HIV, as I have already mentioned, it's a causative agent of this disease, AIDS. So, it results in immunodeficiency. How does it occur? There is a very important cell present in our body that is known as T helper cell. Okay, it's known as T helper cell. What is that? T helper cells are such a kind of cells which help us in providing immunity against foreign pathogens. How? Because these T helper cells actually stimulate the B lymphocytes, which produces, yes, antibody. So T helper cells actually do not produce antibody, right? But they stimulate the B cells to produce antibody. So indirectly, they play a very important role in our immune system. So if something is present in our body that can reduce their number, obviously that will weaken our immune system. So T helper cells, when they are reduced, our immunity will be weakened. So what is the normal count of T helper cell, right? First we have to know this. The normal count is greater than 900 into 10 to the power 6 per liter. So this is the normal count of TH cell, okay? And when AIDS occurs, we may call it full-blown AIDS. When HIV infection occurs, the number of T helper cell might be reduced to as low as less than 200 into 10 to the power 6 per liter. Okay, so you can very easily understand how much weakened our immune system becomes when HIV infects us. And see what happens actually if a person suffers from AIDS now. That person will not die due to AIDS. Yes, because the immune system is so much weakened that any kind of pathogen that normally does not affect a healthy individual will affect these person, because those pathogens are known as opportunists. They are known as opportunists. They are always looking for opportunity. When a person's immune system is weak, they will just jump into that person and they will cause several kinds of difficulties, several problems in that particular person. Like there are a few pathogens like who act as opportunists. Say for example mycobacterium, okay, mycobacterium. Then there is one called toxoplasma. So these are very well-known opportunists which normally infect a person whose immune system is weak. Okay. Now see, TH cell is not the only one which is the target of HIV. Now see, T helper cells are not the only target of HIV. Yes, along with T helper cells, HIV is also target one more cell that is known as macrophage. Macrophage. When HIV infects a T helper cell, it finally kills the T helper cell and thereby reduces their number. But in case of macrophage, the event is bit different, because when HIV infects a macrophage, HIV continues to replicate itself. HIV increases its number, and that's the reason why macrophages are also referred to as HIV factory. This is important for NEET exam. Okay. Now, let me just tell you something which is important for exam point of view, that what are the routes by which HIV infection might occur. See, we always have some misconceptions regarding this. Like very often you might hear that people are very much bothered about some person who is AIDS patient. Right, you cannot touch that person if he or she is an AIDS patient. You cannot shake your hands, you cannot hug that person, even you cannot take care of that person. But these all are myths, because HIV is not spread through physical touch or through mere contact itself. It occurs through body fluids, okay? This occurs through body fluids. So yes, Please be compassionate to those persons who are suffering from AIDS. Please take care of them. Please talk to them. Please give them moral support, because just by mere touch, you won't get this disease from them. Okay, that's just for your information. I think as a part of this society, for being a responsible citizen, we should know this thing. So now I am going to draw the structure of HIV. See, as I have already mentioned that the genetic material consists of single-stranded RNA. So first of all, let me draw the structure of RNA, and yes, two single-stranded RNA molecules are there, and those two molecules are identical. So I have drawn two RNA molecules which are identical, and yes, along with this, let me draw the structure here. Let us assume this is the reverse transcriptase enzyme that I have previously mentioned. Now, this core RNA will be covered by a protein coat. Okay, so let me assume that this is the protein coat, and yes, I am not going to draw the whole part, and this protein coat is known as P24. Fine. Just outside this protein coat, outside P24, there is presence of one more protein coat. Okay, let me draw this. See, this is the outer protein coat. Okay, and yes, this is the coat, and this protein coat is referred to as P17. Yes, so now till not completed. Other than these two protein coats outside P17, there is presence of a lipid bilayer, which is quite unusual. Normally, all the viruses do not contain this bilayer. Okay, so yes, one lipid bilayer will be, yes, one lipid bilayer will be present. Let me just draw two layers. Guys, please don't mind, I am very bad at drawing. Just try to understand what I mean to draw. So this is the lipid bilayer. Okay, this is the lipid bilayer. Now one very pertinent question is, ma'am, from where this bilayer is coming? Yes, it's a very important question. This lipid bilayer is actually derived from the host cell's plasma membrane. Okay, from there this bilayer has been derived. And see, we are not yet done. This lipid bilayer contains some glycoproteins. Okay, so yeah, these are the glycoproteins. Okay, this part is known as pedicel. This is known as pedicel, and we call it GP41. GP stands for what? Glycoprotein. And the head part, this is known as spike. This is known as spike, and this is known as GP120. Okay, so this is our structure of HIV. Okay, yeah, let me just label it. This is single-stranded RNA, and this is the reverse transcriptase enzyme. I think this drawing is understandable, right? Okay. Now see, very important point. This HIV, this virus will affect the host cell. Now, if someone gets attached to another one, there has to be some interaction between those two, right? Now, the spike, that is GP120, this gets attached to the CD4 receptor, which is present on the host cell surface, T helper cell surface. This CD4 receptor will be present, and GP120 will interact with CD4. Okay, this GP120 will interact with CD4. This is again very important for NEET exam. Now, let me just try to make you understand how the HIV cycle occurs, how the replication occurs. Yes, this is HIV replication cycle. What will happen first? See, this is the HIV. Let me assume this is the HIV. So this is the HIV and it contains 2 identical RNA molecules. Fine. Now this HIV will infect yes, this is the host cell. Okay, this is the host cell. HIV will infect the host cell. Let me complete it. Yes, it will infect the host cell. Fine. Host cell. So it will inject its RNA inside the host cell. Fine. This is the RNA. Okay. Now this RNA will be injected along with, yes, reverse transcriptase. Okay, I am writing it abbreviated form RT. Now I have already mentioned RNA in presence of reverse transcriptase will produce, yes, DNA. It will reverse transcribe. It will reverse transcribe to DNA and we call this DNA as cDNA or complementary DNA. Complementary DNA. Okay. Now what will happen? Say for example, this is the genetic material of the host. Okay, this is the genetic material of the host. Now this DNA will be attached to the host cell's genetic material. Okay, so this DNA now gets attached to the host cell's genetic material. Now from this total DNA, normal transcription, normal translation process will occur, right? So from this DNA, after normal transcription, after, I am not writing normal transcription, okay, after transcription will produce some RNA and few of those RNAs will be further translated to make protein and those proteins will make the coat surrounding these RNA. And what will happen? This will finally make the baby viruses. Okay, the baby viruses will be produced. So by this way, the replication of HIV will go on. See how easy it is? Normally we think it's very difficult, right? But actually it's not difficult. But one thing you should remember that retrovirus, HIV specialties, it's a retrovirus and the term retro means back. Since from RNA to DNA, that is why retro, okay? And in presence of reverse transcriptase enzyme, it will make complementary DNA from the RNA. Now this DNA will be incorporated to the host's genome, right? And from there, by transcription, RNA will be produced and few RNA will make protein which will make the coat around the RNA and baby viruses, baby HIVs will be produced. So this is all about our HIV replication cycle. Now a few more important points are left which I think I should mention. So let me just try to mention those points which are important for your NEET exam. As I promised not to take much time of you, so here in this particular board I have mentioned very important points which we will require for NEET exam. First of all, the transmission. First route is sexual route. Like it can occur through multiple sex partner, then by prostitution, then homosexuality or artificial insemination. This might be the sexual route of transmission. Then parenteral route, like in case of blood transfusion. If the transfused blood is not tested, whether it contains HIV or not, then from that particular thing, this disease might occur. Then transplacental route, that means from HIV positive mother to the fetus, it might be transmitted, right? And again, after the birth of the baby, through the colostrum or the mother's milk, it might be transmitted to the baby. Now the treatment procedure. See, there is a misconception like if a person is HIV positive, he is suffering from AIDS. No, it's not that. HIV positive does not mean a person is suffering from AIDS. As I have mentioned, AIDS is a combination of symptoms. Once the pathogen enters a body and the symptoms develop, there might be a long gap and that particular time is known as incubation period. Okay, like the time period from which the virus infects a person and the symptoms appear. So what can be done with these treatments? Like, see, death is inevitable. In all the senses, death is inevitable. But if you take these treatments, you can prolong your life. Like say 20 to 25 years, it can be prolonged. So just be very much serious, very much conscious about everything. Don't die of ignorance. If you suffer from this problem, just go for treatment. Don't be afraid of the social stigma or something like that. So these are the method of treatment. Most important, most commonly used method is azithromycin. It's a medicine, azithromycin or zidovudin. This actually works by inhibiting the reverse transcriptase enzyme. This is RT inhibitor. Then if a person gets say for example resistant to this particular medicine, then DDI or dideoxycyanosin is also targeted. Then if a person is, if a pregnant lady is HIV positive, then doctors very often apply zidovudin and nevirapine to stop the transmission of this HIV from the pregnant lady to the fetus or to the child. So these are the methods of treatment. And finally, the screening. The first method is ELISA, enzyme-linked immunosorbent assay. But mind it, this is important for NEET. This is a non-confirmatory procedure. Confirmatory one is the Western blot technique. So yes, this is all about HIV and AIDS. So as I started by saying this, prevention is better than cure. So yes, please apply your brain and please be compassionate towards those who are suffering from AIDS and don't die of ignorance. Okay, thank you so much.